Hello, AP Biology students. We're going to go over the 10th section in Unit 4. And this section is going to be about the phases of meiosis. Now, one little warning, I started off this unit with 15 sections. I combined through the sections as I was going through this. All the content and information is still going to be there, but I just wanted to make this unit just a little bit shorter and just combine units because I thought the material just went together better that way. All right, with that being said, let's get into what the phases of meiosis are. Within meiosis, there are two divisions. And if you remember with mitosis, there was only one division. So in meiosis, we have meiosis 1 and meiosis 2. Before meiosis 1, normal interphase is going to occur. Remember, that's when the cell is going to prep for cell division and go through replication. It's important we have enough DNA for when the cell splits that each new cell is going to have the correct amount of DNA. Now within meiosis, that's going to be a little bit different since we're going to be going from a diploid cell to a haploid cell. But between meiosis 1 and meiosis 2, there's no interphase. And there's a really important reason why there's no interphase, and I'm going to get to that a little bit later in this section. Another important aspect of learning about meiosis 1 and meiosis 2 is that after meiosis 1, the cell will be haploid. So as you can see here, during meiosis 1, the cell goes from a diploid cell to a haploid cell. That division of chromosomes occurs very early on within meiosis. Whenever the cell splits again, it's going to go from a haploid cell to two haploid cells. And we're going to talk about how meiosis 2 is very, very similar to mitosis. Both meiosis 1 and meiosis 2 are similar to mitosis, so if you understand mitosis, meiosis is going to be pretty easy. What I'm going to do in the next couple slides is go over the major differences between the two. Whenever you look at the individual steps of meiosis 1 and 2, you're going to see that they're labeled prophase 1, metaphase 1, telophase 1, or prophase 2, metaphase 2, anaphase 2. So these phases are denoted with a number that relates to what round of meiosis it's in. So again, I'm just going to go over the differences between mitosis and meiosis in the next couple slides. So in prophase 1, crossing over is going to occur. This is where we're going to see exchange of genetic material between non-sister chromatids of homologous chromosomes. Now there's a lot of words there. I'm going to show you what that looks like here in the next slide. Basically what happens is proteins are going to exchange material between these two chromosomes. Remember that homologous chromosomes have the same genes, but they may have different alleles. And in a pair of homologous chromosomes, one comes from mom and one comes from dad. So now after crossing over occurs, these chromatids are no longer going to be identical. What this does is it increases the genetic diversity in the offspring. Because as you can see in this diagram, at the end of crossing over, you can see that each chromatid is going to be genetically different. This is showing us the process of crossing over, where again, homologous chromosomes are going to pair up and exchange genetic material. Now this image is only showing it occurring once. What actually happens is this occurs many times throughout one chromatid. I'm only showing you this once because it can be complicated looking at a chromosome that's went through crossing over many times. During metaphase 1, another significant difference occurs. Homologous chromosomes are going to pair up along the metaphase plate or in the middle. This is different because before, each individual chromosome aligned in the middle. What's going to happen is something called independent assortment. Independent assortment is when the homologous chromosomes separate independently or randomly from each other. That means the maternal or paternal, the mother or father chromosome, is going to separate from each other and orient themselves randomly throughout the cell. Let me show you what this looks like. You can see in each combination, there are 8 of them. Each combination is going to show a different assortment of the maternal and paternal chromosomes. What this does is it increases the genetic diversity of the offspring cells, or the sex cells that we're trying to produce through meiosis. As the chromosomes line up randomly from each other, again, paternal versus maternal on left or right side, it's going to increase the just genetic diversity or outcomes that are possible. Now for this cell, there are 8 combinations because there are only 3 homologous pairs of chromosomes. You can imagine how many different types of chromosome combinations there are for our cells that have 23 pairs of chromosomes. During anaphase 1, homologous chromosomes are going to be pulled apart, not sister chromatids. If you remember back to mitosis, sister chromatids were ripped apart. During anaphase, a whole chromosome with 2 chromatids is going to be pulled apart. The reason that we are doing this is we are pretty much having the amount of DNA in a cell because we are trying to produce sex cells that are haploid or have half the genetic information. If you think about the goal of sexual reproduction, our sex cells should only have half the number of chromosomes because the other sex cell that it's going to fertilize with is going to have the other half or other set of chromosomes. Now there is a period between the first round of meiosis and second round called interkinesis, but no DNA replications going on. It's just basically a rest period. This is what meiosis 1 looks like. You can see the chromosomes have condensed, they're going to cross over, they're going to align independently during the first couple phases, and then everything kind of looks the same, but remember, these are now still full chromosomes. They're not ripped apart, and at the end here, they're going to be haploid. If you look at the beginning cell in prophase 1, it has 4 chromosomes. Now the cells only have 2 chromosomes. This is because we started with a diploid cell, and now we're ending with 2 haploid cells. Meiosis 2 and mitosis are very, very similar. The major difference is that the starting cell isn't going to be diploid in meiosis 2, it's going to be haploid. But basically it kind of looks just very similar to mitosis because at this point we are going to separate the sister chromatids now. This is meiosis 2, where again, the chromosomes are now going to align in the middle and then be separated, and then each cell will only have 1 chromatid of that original chromosome. Again, in this meiosis 2 process, we are starting and ending with haploid cells. That again should make sense because at the end of meiosis, we need to produce haploid cells or sex cells. I like to use this diagram to represent independent assortment, where our starting cell is going to have homologous pairs of chromosomes. In this case, there are 3. Each of the homologous pairs you can see has chromosomes that are the same length and have the centromere in the same position. Now remember, within homologous pairs of chromosomes, one is going to come from the mother and one comes from the father. Now, in each one of the cells that are going to be produced through meiosis, you can see there is a different combination of the mothers and the fathers. This is because during metaphase 1, the chromosomes are going to align independently on either side, and the result is that each of the cells produced at the end of meiosis is going to have a different combinations of mothers or fathers chromosomes. Now, since there are only 3 chromosome pairs here, again, there are 8 combinations. If we do a little bit of math, we can see that for 1 pair of chromosomes, there are only 2 possibilities. For 2 pairs of chromosomes, there are 4 possibilities. For 3 pairs of chromosomes, there are 8 possibilities. So what we have to do is we just have to put 2 to whatever power of number of chromosomes there are. For humans, if we look at just independent assortment, 2 to the 23rd is over 8 million combinations. So when they say you are 1 in a million, they're not even close because you are 1 in millions and millions and millions. If you're a Rick and Morty fan, this is going to be a great meme for you. I love this. I found this online this year. Meiosis. Well, that just sounds like mitosis with extra steps. Yeah, it basically is just 2 rounds of mitosis, but it's a little bit different.